So hello students and welcome back to another video tutorial by Genome Biotech. In this video tutorial, we would be talking about eukaryotic transcription. Now, what is eukaryotic transcription? Eukaryotic transcription is the process in which the DNA gets transformed to hnRNA and hnRNA gets converted into an mRNA transcript that will be used in order to form the protein molecule. So the protein molecules will be produced by the process of translation through the mRNA transcript that is going to be produced here. Okay. So, what we have to study in the process of eukaryotic transcription, that how does eukaryotic transcription differs from prokaryotic transcription. In case of eukaryotic transcription, the process of splicing takes place, which is completely absent in the case of prokaryotic transcription. In eukaryotic transcriptions, cells have transcription regulation. Transcription regulation refers to the presence of specific molecules that can upregulate or that can downregulate the rate of transcription. In the case of eukaryotes, there are three different types of RNA polymerase involved: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. So, now, firstly, we would be looking at the process of transcription regulation. That how does the process of transcription regulation is going to take place. Then we are going to look at the activities of RNA polymerase 1, which is the RNA that is responsible for the biosynthesis of the mature 5.8S RNA, 18S RNA, and 28S rRNA. So the ribosomal RNA precursor molecule will get converted into 5.8S, 18S, and 28S rRNA. rRNA, that is RNA polymerase second, will produce the product that is in the form of snRNA. Small nuclear RNA as well as mRNA are being produced by the action of RNA polymerase second. Whereas RNA polymerase third is the major enzyme that is required for the production of tRNA, 5S rRNA, and U6 snRNA. These are very important. This U6 snRNA is of the utmost importance. This is the most important U6 snRNA. Another one is the 5S rRNA. 5S rRNA is not produced at the NOR region, not produced at NOR, not produced at NOR, not produced at NOR, nuclear organizing region. It is not produced at NOR. RNA polymerase 1 produces all the different types of RNA except 5S RNA. All the other different types of RNA are being produced at nuclear organizing region. And this nuclear organizing region is present on the chromosome number 13, 14, 15, 21, and 22. These are the region where the RNA polymerase 1 products are going to be produced. RNA polymerase second will produce mRNA as well as snRNA. RNA polymerase third will produce tRNA, 5S rRNA, and U6 snRNA. These are the different products that could be produced. Any doubt, any problem? You just ask. Now, we must also know that RNA polymerase, which is found in the case of eukaryotes, differ significantly from each other on the basis of sensitivity to a chemical that is referred to as alpha amantine. Alpha amantine, which is being obtained from Amanita phyloides. It is being obtained from a fungus that is referred to as the Amanita phyloides fungi. So that Amanita phyloides fungi is responsible for the same. Next, that I am writing, Amanita phyloides. PHALLOIDES. This fungi leads to the production of toxic of alpha amantine, and the RNA polymerase differs in their toxicity to the alpha amantine. RNA polymerase is most sensitive, and polymerase 1 is completely resistant. So the tense sensitivity will follow the following order: RNA pol second being the most sensitive, followed by RNA pol third, RNA pol third, and followed by RNA pol first, followed by RNA pol first, RNA pol first. These are the RNA that shows different sensitivity to alpha amantine. As I was talking about the regulation of process of transcription with the help of upregulation, with the help of enhancer sequences, and downregulation with, that is, reduction with the help of sequences, those which are referred to as silencer sequences. So we have to differentiate how the transcription is going to be regulated with the use of enhancer or the silencer sequences. So let us have a look at the animation shown here. In this animation we would be understanding about the recruitment and increased gene expression, how the increased gene expression is going to take place in the regulation of transcription initiation. In the case of eukaryote, this pathway is exclusively used in the case of eukaryotes. So this is very important. Both transcription transcription regulation transcription regulation is tightly controlled in eukaryote, is tightly controlled in case of eukaryote, is tightly controlled in the case of eukaryotes, in the case of eukaryotes, in order to in order to comply with the needs with the need of the cells of the cell under different physiological conditions. So sometimes the need of the cell requires the high amount of transcript to be produced. At other times, low amount of transcript is being required to be produced. So transcription regulation is tightly controlled phenomena that will regulate the upregulation or the production of transcription in high amount, or the production of transcript in low amount, so that it could be regulated. There are sequences which are present. Now we will have to look at those sequences, enhancer and silencer sequences, as well as the different transcription factors. So let me just start this lecture.
Expression of a gene can be regulated at various stages. The most common is transcription initiation. When you have completed this exercise, you should understand how activation by recruitment increases gene expression, understand how activation by allostery also increases gene expression, and understand how repression decreases gene expression.
Now, as you have been told that the gene expression in an organism is tightly controlled, especially in the case of a eukaryotic cell. So, in the case of eukaryotes, we must know that how to activate the gene expression by recruitment of different activators, how allosteric mechanism is used to activate gene expression, and how repression or decrease of the gene expression is going to take place. So these would be our three aim. In order to understand these three aim, we must go through this animated version of the lecture. Okay, so I'm moving forward.
First, we will review transcription. Transcription is the process by which a DNA template is copied to an RNA strand. The primary enzyme of transcription is RNA polymerase.
So we are going to just review the process. You know that transcription involves the formation of an hnRNA or an mRNA transcript. The major enzyme that is being involved is RNA polymerase. RNA polymerase is going to bind to a specific sequence present over the DNA that is called as the promoter sequences. Because of the presence of -10 and -35 consensus sequences, the Pribno box, the Tata box, the Hogness box, The different types of boxes are present in the case of eukaryotes: the cat box, the up elements, the ntf box, and other different types of boxes or consensus sequences are present over the promoter sequence. That will be the site for the binding of RNA polymerase enzyme. Moving forward.
Transcription initiation begins when RNA polymerase binds to a region at the start of the target gene called a promoter.
Now, it is here from the transcription initiation is going to begin. The RNA polymerase is going to bind to a specific site over the DNA, and this specific site is commonly referred to as the promoter sequence. A region at the start of a gene where RNA polymerase bind and initiate the process of transcription is referred to as the promoter region. So, promoter is just a region at the start of the gene where RNA polymerase molecule is going to bind, and then it is going to initiate the process of transcription. How?
The initial RNA polymerase promoter complex is called a closed complex because the DNA strands remain together.
If you have any problem, just go to the previous lecture. In that lecture, I have described the entire process of transcription. In this lecture, we are going to focus upon regulation of transcription initiation. That's all.
RNA polymerase then causes the DNA at the start site of transcription to unwind. The complex in which the DNA is unwound is called an open complex. In the final step of transcription initiation, RNA polymerase leaves the promoter and begins transcribing the gene. This step is called promoter escape. Many promoters are imperfect. In the absence of regulatory proteins, RNA polymerase binds only weakly.
Now, there are two different types of promoters. One type of promoters are the one which are very strong. They have the ability to themselves bind to the promoter and initiate the process of transcription. They do not require any help from other. Okay, such type of promoters are called as strong promoters. Whereas there are other different types of promoters that requires the help of other different enzymes that should help the promoter sequences for binding with the RNA polymerase. Such type of promoters are called as weak promoters. So there are basically two different types of promoter. One is said to be the strong promoter, another one is said to be the weak promoter. Now these two promoters differs from each other on the basis of their activities. Moving forward.
Occasionally, RNA polymerase is able to successfully bind to these promoters and produce an open complex, leading to a low level of constitutive expression called the basal level. RNA expression can be increased over the basal level using activators, or decreased using repressors.
Now this is the key important point. You should know that there is a basal level of transcript of every gene present at every location in your genome. What do you mean by basal level of transcript? The concept of basal level of transcript state that it is the amount of RNA molecule expressed without the action of regulatory proteins. So without the action of regulatory protein, how much amount of transcript is going to be produced? That is said to be the basal level of the transcript. So this is the minimum level of transcript. This is called as the basal level of transcript. The minimum amount of the transcript that would be produced without the act of regulation is called basal level of transcript. So I'm just writing it out. The minimum amount of the transcript, amount of the transcript constantly being produced, the minimum amount of the transcript constantly being produced without the involvement of any regulatory protein such as enhancers, silencers. Regulatory proteins will be activators, co-activators, activators, co-activators, repressors, etc. Thus, such type of such type of DNA is constantly expressed. So such type of DNA is being constantly expressed, then this is referred to as the minimum basal level of the transcription that is going to take place. So this is referred to as the basal level of transcription. Now, there are other different types of genes that are always expressed. Those genes which are always present in the expressed form are called as constitutively expressed genes. Now, these genes are the one without which without the action of regulatory proteins, they are always being expressed. Now, when you are going to talk about genes, the genes can be of two different types in the case of Homo sapiens. The first type of genes could be housekeeping genes and another could be luxury genes. So we can classify the genes into the following types. One is referred to as the housekeeping genes and another one is referred to as the luxury genes. Okay. So there would be I would be talking about the first one, housekeeping genes. What do you mean by housekeeping genes? Housekeeping genes are the one that are constitutively expressed. These genes are constitutively expressed. These are constitutively expressed. These genes are constitutively expressed. Constitutively expressed as they are essentially required for the sustainability of the cell. As these are essentially required. These are essentially required for the sustainability of the cell. Required for the sustainability of the cell. Sustainability of cell. These are required, essentially required for the sustainability of the cell, and such type of genes are referred to as housekeeping genes. Another one is referred to as luxury genes. What do you mean by luxury genes? Luxury genes के अंदर क्या हो जाता है? What are these luxury genes? Luxury genes are the genes. They are also referred to as smart genes, and these genes are functional only when required. These are functional only when required. These are functional only when required. Functional only when required. Okay. All the different types of genes that are regulated. For example, you can, in the case of prokaryotes, such type of genes are referred to as operons. So whether you talk about the lac operon, you talk about TRP operon, all such type of operons are the example of luxury genes. Now, the third type of genes that are referred to as the regulated genes. Now, they could be the third type. The third type would be referred to as the regulated genes. What do you mean by regulated genes? The regulated genes are the one. Regulated genes are the one which are always expressed at their basal level. These genes are always expressed. These genes are always expressed in their minimum amount or basal level, and can be upregulated or downregulated. So these genes can be upregulated or their downregulation is also present. Then such type of regulation takes place in the case of regulated genes, and such type of genes are commonly referred to as the regulated genes in nature. So such type of genes are called as regulated genes. Next point that we are going to talk about. Now, the regulated genes can be positively regulated, they could be negatively regulated. Okay, so these housekeeping genes are always constitutively expressed, whereas regulated genes are always expressed at their basal level, and they could be activated with the help of activators and co-activators, or could be repressed with the help of repressor molecules. So these two molecules will antagonistically control the activity of these regulatory genes. Okay, so we are going to move forward.
An activator is a regulatory protein that increases the level of transcription over the basal level. Activators typically bind to a site on the DNA near the promoter.
Now, there
Most
are there are certain proteins that will really activate the process of transcription, that will enhance the process of transcription. Such type of proteins are called as activator proteins. Activator proteins will bind to a specific site over the DNA, and that specific site is referred to as activator binding site. This is the site that is present where the activator is going to bind. Once the activator will bind, it will cause interaction with the RNA polymerase and will cause the RNA polymerase to bind to the promoter sequence. Now, how this process takes place? Now, our promoter is a bit shy. He cannot interact with any RNA polymerase. So, what the problem is that the promoter is a least interested person. Okay, he's basically a boring person, a weak promoter. Okay, this is a weak promoter. Once this promoter is a weak promoter, then this promoter is always going to hesitate while interaction with an RNA polymerase is going to take place. Okay. So what happens? The promoter is not able to bind to the RNA polymerase. Then activator, he calls upon his friend. Oh, hello activator, how can you help me? Can you help me in binding with the RNA polymerase? This RNA polymerase looks so cool. Can I bind with this RNA polymerase? Okay, then the activator says, okay, I will use three-step strategy to make you bind to the RNA polymerase enzyme. Okay, what are the three steps strategy? So the three-step strategy state that RNA polymerase will be told to spend some time with the promoter sequence. The activator will cause the RNA polymerase to spend some time with the promoter sequence so that the effective interactions can take place. And once the interaction will take place, the bonding will take place. How? Look here.
activators have two binding sites, one for interaction with DNA and one for interaction with RNA polymerase.
So, see this. Promoter was interesting in binding to RNA polymerase. Then promoter called upon his friend activator and they made a plan. Promoters told activator that I am going to throw a party. You bring RNA polymerase with yourself and kindly introduce RNA polymerase to me, that I am known as promoter. The name that you would have always heard. The name was Raj Malhotra. So similarly, promoter is going to bind with the RNA polymerase. How? Actually, activator is the one.
Typically, activators
Activator is the one that is going to enhance the process of transcription. See, one polymerase came because of activator, transcription is taking place. Here transcription will take place, but it is very slow, very slow. See, in the presence of activator, what is the amount of transcript? 4 transcript, 5 transcript, 6 transcript. Every time the transcript is coming and the transcription is going on, the RNA polymerase is coming, there is no RNA polymerase that is left, and promoter is going to transcribe every gene. And here, what is going to happen? The promoter is unable to transcribe the genes because this promoter was unable to interact effectively with RNA polymerase. See, RNA polymerase came and then went away. But here, see how much is the amount of transcript produced? A very large amount of transcript is going to be produced without activator and with activator. So, what the activator did was something like this.
increase RNA expression by binding to the DNA and RNA polymerase simultaneously and holding RNA polymerase in place long enough to initiate transcription. This mechanism of activation is called recruitment.
Now, This is the mechanism of activation that is going to be taking place. Here, the activator recruits the RNA polymerase and makes the RNA polymerase to bind to the promoter. Once it binds to the promoter region, now then the process of transcription is going to be initiated. A mechanism of activation in which the activator simultaneously binds to DNA and RNA polymerases holds the RNA polymerase in place to initiate the process of transcription.
Not all promoters are activated in the same way. Sometimes RNA polymerase is able to bind to the promoter efficiently, but the RNA polymerase promoter complex isn't able to efficiently convert to the open complex to initiate transcription.
So sometimes the RNA polymerase is able to bind to the promoter, but it cannot move to the open complex and cannot initiate the process of transcription. Then what is going to happen? How the process of transcription is going to be initiated in this reaction mechanism? The activator will come here. How?
At such promoters,
This is the first mechanism. Okay. Sorry.
activation is accomplished by allostery. Activators interact with the stable closed complex and induce a conformational change that causes transition to the open complex.
See, binds causes the interconversion of closed complex to the open complex. Now, promoter was suffering from a problem, although he was able to interact, but the closed complex was not getting converted into the open complex. The things were not getting on. Every time the promoter binds to the RNA polymerase, the RNA polymerase always says, I have never thought about this. Okay, so a promoter was a bit frustrated. He called upon activator, and activator solved the problem by making the promoter bind to the RNA polymerase. And this is the amount of transcript. A very high amount of transcript is being produced in the presence of the activator molecules. Okay. Now, next point.
Some activators that act by allostery induce a conformational change in RNA polymerase.
So there are two processes that are going to take place. Either the RNA polymerase will undergo conformational change, or the DNA must undergo conformational change. So the first mechanism is this. Some activators that act by allostery induces a conformational change in RNA polymerase. So here there is a change in the RNA polymerase that has been brought together. Now moving forward.
While others induce a conformational change in the promoter DNA.
See this. Here is the conformational change in the DNA itself. Process of transcription is going to take place rapidly. The third process. This is basically allosteric mechanism only, in which the either the DNA has been changed or the RNA polymerase was being altered.
A repressor is a regulatory protein that decreases the level of transcription.
Now, that one was referred to as activator, means good friends. And this is referred to as repressor, means best friends. Now, what the best friend is going to do? Okay.
The site on the DNA where a repressor binds is called an operator.
So, okay. So the repressor is going to bind with the promoter molecule in such a manner that promoter cannot interact with any RNA polymerase. Whenever repressor is present, there would be no RNA polymerase that will have the ability to bind with the promoter sequences. How? Repressor binds to a particular site in the promoter, and that is referred to as the operator site. Now see what is going to happen.
Repressors often bind to a site on the DNA which overlaps the region bound by the RNA polymerase. When
Okay,
a
so there is an RNA polymerase that is going to arise now here. Okay, so once the RNA polymerase arises, okay, there is an RNA polymerase, and this RNA polymerase is looking to bind to the promoter region. It went here, but what happens? The repressor is already bound. Okay, what will the RNA polymerase do when the repressor is bound to the promoter region? The RNA polymerase has no chance of binding. It cannot bind. It asks, can I bind to the RNA polymer to the promoter region? Repressor says that the promoter is only mine. You cannot bind it. So promoter is going to bind only to the repressor region. They are in a relationship, so this cannot bind. The RNA polymerase cannot bind, and the RNA polymerase is going to move away. It is going to move away. There is no binding. As there is no binding, so rate of transcription is decreased. Rate of transcription is drastically decreased. This is referred to as repressor molecules. Okay, look here.
When a repressor is bound, the RNA polymerase is unable to bind, thus decreasing or eliminating RNA expression.
So RNA has been eliminated. RNA expression has been eliminated.
Not all repressors act in the same way. Some repressors inhibit transition to the open complex.
Some of the repressors molecules are such that even if the promoter is going to interact with the RNA polymerase, they will inhibit the entire process and they will spoil the entire process. These are referred to as the best friends, the friends of the friends. Those are referred to as repressor molecules. Even an interaction is going on, they will be coming and interfering. See this?
Other repressors inhibit promoter escape.
Other repressor, it is inhibiting the promoter escape. Even the things were going to go on and the repressor has inhibited the promoter escape. The promoter and RNA polymerase were going to go on, the repressor came in between and pulled the RNA polymerase backward so that there is no active interaction to take place, and ultimately the RNA polymerase will be lost.
Regulatory proteins are often controlled by the substances which the target gene's protein product metabolizes, or by related substances. This exercise will help you understand how this regulation occurs.
Now the exercise is the most important and the most tedious work to do. Okay.
Bacteria has been discovered that is capable of metabolizing a rare sugar that is produced by the flowers on which the bacteria live. This bacteria prefers to use glucose as an energy source. However,
There is a bacteria that is living over the flowers, and this bacteria has the ability to use glucose as a metabolic source of energy. Now,
when glucose is not available, the bacteria switches on the genes for the production of the proteins to metabolize the rare sugar.
a bacteria is able to metabolize a rare sugar, and this rare sugar is present in the case of flowers. The first substrate for the bacteria is glucose, and the second is the rarest sugar. Without glucose, genes for rare sugar metabolism are switched off. Only these genes will be switched on when there would be no glucose molecule.
You have isolated the gene and promoter for one of these proteins. We'll call the gene ALT1. You can use a northern blot to determine the level of RNA expression for this target gene and DNA footprinting to identify the locations where proteins bind to the DNA.
Okay, there are two techniques that has been used. One is the northern block, another one is the DNA footprinting assay. The DNA footprinting assay will be used in order to depict where the proteins are going to bind, whether the enhancer is going to bind or whether the repressor sequences are going to bind. The northern blot will be used in order to see that what is the amount of transcript that is going to be produced. And we have the gene that is ALT1. This gene will be transcribed only when the glucose molecules will be absent. So let us look at the first question.
The expression level of the gene changes depending on whether glucose and the rare sugar are present. Select whether glucose and the rare sugar are present when the bacteria are grown and press Display Results.
Okay. When it is available and when the sugar is not available, then what is going to happen? Then this gene will not be transcribed. This gene will not be transcribed. You cannot transcribe this gene because the glucose is present and the rare sugar is said to be absent. Select whether glucose and the rare sugar are present when the bacteria are grown and press display result. Select whether glucose and the rare sugar are present when the bacteria are grown and press display results. Display results. Okay.
When the bacteria are grown with only glucose, ALT1 RNA is not expressed.
I've
DNA
already told you that when glucose is there, then this is going to inhibit the alt1 RNA. Okay, alt1 is the RNA, is the gene which is required, whose protein product is being required for the metabolism of rare sugar. It is going to produce an enzyme, and this enzyme is going to metabolize the rare sugar. It is going to metabolize rare sugar. Metabolize the rare sugar. Next point.
DNA footprinting indicates that a protein binds within the promoter DNA.
Now, DNA protected by proteins. DNA footprinting assay is going to show that the protein is going to bind within the promoter region of the DNA. That means in the presence of glucose, the protein that is being bound to the promoter region is going to be the repressor molecule. Any protein that is going to bind within the promoter region will be referred to as the repressor. So this would be the repressor. It is going to bind within the promoter region. When it is going to bind within the promoter region, this gene will be referred to as the sorry, this gene will be referred to as the repressor gene. Okay, just a second. Okay, this would be repressor, as it is going to bind within the promoter region. Next point. Now what? I have to make the other things available. Okay, now what? When the glucose is available and the rare sugar is available, still the glucose will not allow the rare sugar to be transcribed. The same results will be obtained. Display result.
When the bacteria are grown with both glucose and the rare sugar, ALT1 RNA is expressed at a low level, and DNA footprinting indicates that only RNA polymerase binds to the DNA near ALT1.
So here, alt1 will be expressed at a very low level, and this low level will be referred to as the basal level of the transcript. It is going to be expressed at a very low level called as basal level of transcript. Basal, BASAL, basal level of transcript. Level of transcript. Next point. Now, when the glucose will become not available and the rare sugar will become available, then the process of transcription is going to take place at a much faster rate. The alt1 will be expressed at a very high level. See,
When the bacteria are grown with only the rare sugar, ALT1 RNA is expressed at a high level.
why? Because here the enhancer sequences or the activator proteins will be biosynthesized. Here the proteins that will bind will be activators, and activators would have enhanced the binding of the RNA polymerase. So I'm just showing you here activator proteins also. Please look here. These are the activator proteins which are present, and these activator proteins are going to bind. Once the activator proteins are going to bind, then they will activate the rate of transcription from this particular RNA molecule. Okay, next point.
DNA footprinting indicates that RNA polymerase binds to the promoter, and another protein binds upstream of the promoter.
So there is another protein that is going to bind to the upstream of the promoter DNA sequences. Next point. The next question that is going to come here. Now, the last one, when both are not available, nothing is going to happen when both are not available.
When the bacteria are grown with neither glucose nor the rare sugar, ALT1 RNA is not expressed.
It will never be
DNA
expressed.
footprinting indicates that a protein binds upstream of the promoter, and another protein binds within the promoter DNA.
Okay, next point.
Question 1. For the instance where both sugars were made available, Select which proteins bind to which sites.
Now, which protein is going to bind to which site over the DNA? For the instant where both sugars are made available, select which protein is going to bind. The RNA polymerase is going to bind to this particular site. Okay. And then for which protein, when both the sugars are being made to being available. Okay, small amount of transcript has to be produced. So we are going to bind the RNA polymerase molecules here. Which protein will bind to which site, and the repressors will be binding to this site, so that the process could be repressed, but still the basal level of transcript is going to be taking place. So why there are four molecules being given here? Okay, let me check.
Incorrect. You may want to
Let me see this, what they wanted to say. Because RNA polymerase is going to bind to the promoter region, and this would be the promoter region, but there might be no repressor.
correct.
Okay, because why? Because see, they are saying that glucose is available and the rare sugar is also available. Because rare sugar is available, so some amount of transcription is going to take place. So basal level of transcript will be produced. The basal level of transcript will be produced when there will be no repressor present. So it will produce the basal level of transcript. Basal level of transcript will be produced. Okay, this would be the lowest or the minimum level of transcript that would be produced and is referred to as the basal level of transcript. Next point, next question.
Question 2. Select which proteins bind to which sites when only glucose is present.
When only glucose is going to be present, only glucose molecule, then the repressor is going to be bounded. Because see, here the DNA footprinting assay shows that no protein is binding. There is only a single site where the proteins could bind. And where this protein could bind, which protein could bind when the glucose is present and rare sugar is absent, then the repressor will be binding to this particular site. Okay, any other site? Okay, just a second. Okay, this has been bounded.
Correct.
Okay, so repressor binds to this particular region in order to inhibit the process of transcription when glucose is available and the rare sugar is not available, when the rare sugar is absent and glucose is present.
Question 3. Select which proteins bind to which sites when only the rare sugar is present.
When only the rare sugar is present, then the gene will be highly active. That means the enhanced rate of transcription is going to take place. And the proteins that could enhance the rate of transcription will be the RNA polymerase activated with the help of a protein that would be referred to as activator. So, activator is going to bind here and it is going to induce the RNA polymerase to be activated for the process of transcription.
Correct. Question 4. Select which proteins bind to which sites when neither sugar is present.
Now, no sugar is going to be present. When there is no sugar transcription that is going to take place, then which protein is going to bind to which particular site? Okay, there is no sugar present. So when there is no sugar, the gene cannot be transcribed. You cannot transcribe the particular gene. So the repressor molecule is going to bind to this particular site and it is going to repress the DNA transcription that could take place. Okay, and then it would be completely repressed. The other site will be the site for the binding of the repressor or the activator sequences. Drag the protein to the site where the protein binds. You must not need all the 5 proteins. Okay, so this would be the site for the binding of activators. I didn't know it can bind to this particular sequence, or it could also. Okay, let me check. This would be the most appropriate one.
Correct.
Okay.
Question 5. Which step of transcription initiation is most likely inhibited by the repressor?
Now, how the repressor is going to inhibit the process of transcription initiation? Which step of the transcription initiation will be inhibited by the repressor molecule? Okay. So transcription initiation. When both are inhibited, then RNA polymerase binding to the promoter will be inhibited. Transition, this first step will be inhibited. When first step, when there would be no RNA polymerase
Correct.
binding to the promoter, then the process of transcription will itself be inhibited. Okay.
Question 6. How is the repressor regulated?
How is the repressor going to be regulated? It is the glucose molecule that is going to turn the repressor on, or glucose turns the repressor on. The rare sugar turns the repressor on. The rare sugar turns the repressor off. So it is being regulated in a manner. The rare sugar is going to turns the repressor off.
Correct.
Okay. So rare sugar, when will be present, it will turn off the repressor molecule.
Question 7. Which mechanism of activation is most likely used by the activator?
Now, which mechanism of activation can be used? It can use either mechanism. So we must perform further experimentation
Correct.
necessary to determine whether it could be recruitment or whether it could be allosteric mechanism of regulation.
Question 8. How is the activator regulated?
How is the activator regulated? The activator will be regulated when the rare sugar is going to turn the activator on. Okay.
Incorrect. You may want
Just a second. The rare sugar will turn the activator on. The glucose turn the activator off. The glucose turn the activator off. Okay. Glucose is going to turn the activator off. In the absence of the glucose molecule, the activator is going to be turned on. Sorry. In the absence of the glucose molecule, the activator is going to be turned on, and then the process of transcription is going to take place.
correct.
Okay.
In the absence of regulatory proteins, RNA is often expressed at a low basal level.
This is a very important conclusion, because this conclusion helps to explain that how in the case of lack of operon, the permease enzyme is going to be expressed when the lactose will be present. Now, the last point that I had to extrapolate in front of you is that I'm making a beaker in front of you and trying to answer a very important dilemma of molecular biology. For example, this is a conical flask, and within this conical flask you are going growing E. coli culture. This is the conical flask, and you are growing E. coli culture within this conical flask. Now, within this conical flask you have glucose molecules, and along with the glucose molecule you have lactose. Okay. The primary carbon sugar for the E. coli, I'm showing E. coli with red color. These will be the E. coli cells. E. coli cells. Now, E. coli cells will have different types of components. See, I'm writing here the primary carbon sugar, the primary, or bracket 1 degree. This is first carbon sugar, carbon sugar for the E. coli, for the E. coli. E. coli bacterium is always glucose, followed by by lactose, which serve as the secondary carbon sugar, followed by lactose, which serves as the secondary carbon sugar, carbon sugar. Okay. This lactose, this lactose will be used only when this lactose will be used only when the glucose is absent. Glucose is absent in nature. Okay. It would be used only when the glucose is absent in the solution, in the solution, or when glucose has been completely used, has been completely used or metabolized or metabolized. The problem that I'm going to ask you here is that this is a very, very important problem. Now, supposingly, if the glucose is metabolized, glucose is completely metabolized, the bacteria under culture, the bacteria under culture will exhibit the dioxic growth and will now use the glucose molecule, now use the lactose molecules instead of glucose. But as the lac operon is completely inactive, so lactose cannot be utilized. Do the bacteria will die? This is the question. See, previously glucose was present in abundant amount in the cell, okay, but later on the concentration of glucose began to decrease when it was being metabolized by the bacterial cells. And at after some time what is going to happen? After some time, glucose is going to be eliminated. Glucose will be eliminated, and only lactose will be present. Only lactose will be present. It is the lactose molecule that would be present here. Okay, but because the lacoperon, as you all know, that when we are going to talk about the lacoperon, it has three different genes. Which are the three different genes? The lac, Z, Y, and A are the three different genes which are present. Now supposingly, if this is the promoter region, that is going to be bounded. Sorry, I'm going to make this with another color. Sorry for the disturbance caused here. Let me just draw this. Just a second. Supposingly this is the promoter region. Okay, then after the promoter region you have the structural genes present, and those structural genes are, this is the first structural gene, Z. Then you have the second structural gene that is referred to as Y, and the third structural gene that is referred to as A. These are the three structural genes that are present. So here actually the promoters will not be able to be, this is the promoter region, this is the promoter. These are the genes which are present: lac Z, Y, and A, lac Z, lac Y, and lac A. This is beta galactosidase, beta galactosidase. This is permease, and this is transestylase. Transestylase. Okay. Now I'm saying to you all that when the lactose was absent, then when the glucose was present, glucose was present at high concentration, no transcription was taking place, no transcription. Okay, but what happens here is that instead of no transcription, actually low transcription was taking place, and such type of low transcription is called as basal level of transcript. This basal level of transcript means that when the glucose will be finished, when glucose will be finished, glucose will be depleted, will be depleted, cell will have cell will have the permease enzyme in order to increase the permeability of the lactose. Cell will have the permease enzyme to enhance lactose entry into the cell. Lactose entry into the cell. Entry into the cell. Into the cell. So the concept in the question, see this, the culture exhibit dioxide growth. So now use the lactose molecule instead of glucose. So the culture is going to use the lactose molecules instead of glucose. But as lac operon is completely in active state, so lactose cannot be utilized. This is wrong. Lac operon is not in its inactive state. In fact, lac operon is made always to be present in its basal level of transcript. Lac operon, when glucose is present at high amount, when glucose will be depleted, cell will have the permease enzyme to enhance lactose entry into the cell, so that the survival of the cell can take place. So that the survival of cell can take place. That the survival of cell can take place. Okay. So here the survival of the cell can take place. This is how the survival will take place. So that's all for today's lecture. I do hope that you enjoyed the lecture. You were able to gain knowledge. In tomorrow's lecture, we would be talking about the process of transcription from Pathfinder, and we would be looking at the eukaryotic promoter sequences, which are the different types of promoter sequences for RNA polymerase first, second, and third, and finishing the basal level of transcription. This is the point that we have made clear, that how the activators and the co-activators are working, in what manner these proteins work together along with the repressor protein. So that's all for today. Hope you enjoyed the lecture. Thank you. Over and out. Thanks a lot. Thank you. Good night.